virus is big enough to span so many countries? No. What is it that is happening?
Everyone's dying. It's spreading really fast.
Spread an infection that goes throughout many. Okay, your description was a good thing. So it's infections that are spreading throughout multiple countries and it could be across the world. Basic thing about a pandemic is that it is not localized or held in one area. Oops. Froze there. What things would be common between all types of viruses? Cedric, what do you think?
Environment,
Huh?
like the three things, adapts environment, group and develop, and metabolism.
Okay. Metabolism.
Yeah.
So, all right. I see where you went there. I guess I was looking more for like structure-wise, physically.
Yeah, I did what you did.
I also did what you did. They all have a little outside of
Capsids.
Yeah, I'll have capsids. Okay.
Yeah.
So yeah, if you put the features of them, I guess that's okay. But let's look at the physical aspects. So they got a capsid.
DNA or RNA.
They have some sort of genetic material. That's about it. They're pretty simple overall. So sorry about the vagueness of the question there.
Oh my god, I need to energy. No. Because no. Oh my god.
Okay. Casey, what would a bacteriophage be?
Virus that infects bacteria that you can get. Yeah, I don't know.
That you can get. I don't think you need the last part. First part was fine. Yeah, bacteriophage is a virus that can infect bacteria. So they use bacteria as their host cell. Okay, what is the function of a glycoprotein on the outside of a virus?
It's an ID checker.
ID checker, yeah. It's for recognition, say.
Yeah.
You can think about it as sort of like a license plate or something like that. It basically gives a way for the virus to kind of have a specific part of itself that would be different than any other virus. That could be useful for integrating with the host cell. Okay. Number 5, what observations support viruses as being alive? What sort of things would we point at if we were trying to make the argument that viruses are living organisms?
They evolve.
They evolve? Anything else?
They
They adapt to their environment? Order, non-random structure? You said they adapt to their environment.
Metabolism.
Yeah, the metabolism kind of goes a little bit both ways. They don't really use their own energy source. But those would be the main things. They adapt to their environment, they have an ordered structure, and then they evolve. Okay, what things would we look at to say that maybe they're not really alive? Yeah, they don't really have homeostasis. They cannot reproduce or grow and develop without help of a host cell. That is really the main one that we look at as far as viruses not being able to be alive. They don't really have metabolism in the sense that they don't use their own energy. They're more passive. Okay, what would be a viroid, Austin?
A virus. A virus like chunk of naked DNA that can affect cells.
Okay, pretty good.
Okay.
So it's like a virus, but there's no capsid, there's no glycoproteins, there's no envelope. It's basically just DNA or RNA that, if it gets inside of a cell, can cause problems. But the biggest thing is it can cause the reproduction of more viroids. Okay, what's prion? No, this is not saying general DNA that affects them. It's a viral.
I'm number 8 now.
God dang it. This isn't my day.
Sorry. So it's a misfolded protein. That's not all it takes to be called prion.
It causes others to.
It can cause other proteins to misfold, and that's important. Misfolded proteins do show up. They can kill cells and make things so they don't work right, but they usually just cause that one cell to die. No big deal. The big thing about these diseases is that they can be passed on to other cells and other organs, so they can cause other things to misfold. Okay, here's my Lytic cycle. If you don't have exactly what I have, it's wrong.
Thanks, Mr. Roark.
Just kidding. The basic things that you need to have are the infection. So this virus has to get its DNA or RNA into the cell in some way, and then reproduction of that virus material, and then the breakout of the new viruses to be able to go on to infect other cells.
That's exactly what I had. Because you didn't label it. Who labels really? That's like barely labeled. There's like
Yeah.
10 words.
Well, yeah.
At least it's
But on a test, you should label what's going on.
well. People who say people who built the so you or set goals.
Okay, and then how do viruses reproduce during the lysogenic cycle? When the cell copies itself, when it goes through DNA replication and reads the DNA strands and makes new copies of them before it's going to split. Because that virus DNA is in there, that gets copied along with it.
DNA with that blue.
Cell copies the virus DNA along with its own.
We done for
It is Friday.
Today we're going to talk about bacteria.
Yeah.
Yes!
Yum.
I just feel like
Okay. Now, bacteria are definitely considered living things. They exhibit all of the processes that living things do, so there's no debate on it like there's with viruses. They are actually more complex than viruses are. Now, different bacteria cells will have different features. These are the common features to all bacteria cells that we're going to talk about. But you may see some different things, and we'll talk about things like flagella and cilia and that kind of stuff when we look at other bacteria cells. So, any bacteria cell, it has to have some genetic information, so that is going to be in the chromosome.
So we moved on from viruses.
Yes. Okay? In a bacteria cell, the chromosome is going to be one long circular strand. Now here you can see it's all kind of bunched up. So just like if you take a rope and you just throw it in the corner, if you take a rope that's tied to its own end and you throw it in the corner, it could be all on top of itself and stuff. But if you were to untangle that and pull it out, it would be a big circle. And that's where all the genetic material is in one chromosome. So they don't have 23 pairs like we do. They've just got a single chromosome. But they do have some smaller chunks of DNA called plasmids. Those are small circular parts of chunks of DNA we're going to have. It's not going to be the genetic information for the cell, but there is going to be some genetic information for like making ribosomes and things like that out of plasmids. So,
Together, awful.
got a chromosome, you got some small plasmids, that's all the genetic material. But of course, for that chromosome to be useful, we have to be able to put together proteins. The whole point of the chromosome is to code for proteins, so we need a place for proteins to be made, and those, of course, are ribosomes. So if you remember back when we studied cells, we talked about prokaryotic cells have ribosomes along with the eukaryotic cells. I was just waiting until we talked about it. So, all cells have a plasma membrane. Bacteria cells are no different. It's much like the plasma membrane that makes up our cells. So there's a lipid bilayer. And that's what this kind of lighter yellow colored thing is showing here. What?
Whatever it before.
Plasma membrane?
Oh, lipid bilayer.
Yes. Remember the lipid bilayer? There's the phospholipids that have the hydrophobic heads and the hydrophilic heads, hydrophobic tails. They stick together like this, right? Okay. But bacterial cells have something that we don't. They have a cell wall. Now this wall is going to be a little bit different than the cell walls in a plant cell, but it serves the same function. So it's made out of a material called peptoglycin. And the whole point of the cell wall is to maintain the shape. Water is continuously rushing into the bacteria cell because of osmosis. Osmosis.
Yeah.
So because there's stuff in here, right? There's ribosomes and sugars and all sorts of stuff in here. The water is trying to balance it out, so the water is continuously rushing in. And that means that the bacteria cell has a pressure kind of pushing out because it's continuously kind of being filled with water. Without the cell wall, the bacteria cell will explode. We exploit that fact through antibiotics. When we introduce antibiotics into our system, the antibiotics actually put holes and break down the cellular wall so that the water that's rushing in explodes the bacteria. We talked about this before.
Okay.
And that's the basic makeup of a bacteria cell. Chromosome, plasmids, cellular membrane, cell wall, ribosomes.
DNA was messed up.
Kind of all strung up. Yes, Marley.
So if you get the wrong antibiotics, then it won't put holes in them.
Right. There's different bacteria strains. We'll talk about that in a little bit. And so they will, depending on the type of bacteria strain, they'll give you one kind of antibiotic versus another. There's ways, and we'll look at one way that you can tell kind of what family of bacteria you get. Now, as far as bacteria cells go, they do come in different shapes. So there's three main shapes. One looks like rods. We call them bacillus. So this is a shot through just a normal microscope. You can see the rod-colored shape there. This is through an electron microscope. So the capsid, or not the capsid, sorry, the cell wall and the plasma membrane just form a shape of a rod. All bacillus family bacillus of bacteria are going to have this shape. Okay, some form tiny little balls, orbs. We call those coccus. Right.
I was gonna spell it for you.
All right, control yourself. And the third family are spiral-shaped bacteria, which we call spirillum. Okay, so the shape can help kind of classify what type of bacteria you have. We can see that just by looking under microscope slides at the bacterial cells. Now, some bacteria, not all, but some, are able to create a hard outer coating called an endospore. And they'll create this endospore when the environment deteriorates. Bacteria like environments that are warm and moist. Well, mold is a completely different organism. But they do tend to grow in similar situations. For example, one place that it tends to be warm and moist is your armpits. What? So if you don't shower for a while, the bacteria colonies that grow in your armpits will multiply, and they give off a certain odor. And that's what BO is. So when you smell BO, it's not actually body odor. It doesn't come from your body. It comes from bacteria that are growing on your body.
So everyone take a shower, please.
And you can get rid of most of the bacteria by washing them off. You're not going to get rid of all of them.
But it's on your back.
What's that?
But it's on your back, so that's like.
What? But it technically, it comes from bacteria.
So really, we should wipe our armpits with Germ-X, because it kills 90% bacteria.
Will that help, actually?
I feel like it gets sting.
It's going to sting. It's not probably going to help really all that much because that Germ-X stuff, yes, on contact it may kill a bunch of the bacteria, but it evaporates like that, and once it's gone, then plenty of spot for new bacteria to roam in, to come in and start colonizing. There's bacteria everywhere.
They evolve so that they're used to it.
They could.
So what if you take a shower and like 2 hours later you stink? Does that just mean bacteria is growing?
Pretty soon we spit in our armpits on each other.
Could mean that you didn't wash yourself good enough.
Disgusting people.
Hey, we wear hats.
Now, this means that some bacteria can basically hang out anywhere until they find the right environment. So like the whole 5-second rule, yeah, that's really dumb. As soon as your food hits the floor, there are bacteria endospores on it, and they will.
Yeah, I dropped my pizza on the floor and it was in the bar. And I could have wasted it, so I feel like crying after
The
it.
bacteria endospores will stick to the food right away. Doesn't matter how long it's there. It's not like,
I'm gonna die.
listen, it's not like the bacteria are going to be crawling towards the food trying to get there. The point is, your food falls on and they stick to it. You put it in your mouth, ooh, here's a good environment to grow. Hey, the
And then
They're growing in your body, right?
endos, the endospore breaks down, the bacteria starts to live and divide, and you get a bacterial infection. Yes?
Doesn't it like build your immune system though?
There is that. If you keep things too clean, especially like when you're raising your children, if you keep things too clean and they're never exposed to anything, then they can have an immune system that it's basically not up to the fight if they ever do get an infection. So, I mean, I don't know.
Make sure your house is dirty.
You can definitely be way too clean and too antiseptic about stuff, but I also wouldn't necessarily eat pizza off of the bar floor. So if you want to find a happy medium,
If you like drop your cookie on the floor,
At the bar, guys.
throw it away.
I don't think it matters what I would do.
At the bar, and what I mean is
That place is dirty.
you if you watch the documentary on the floor. Like if you
Yeah, I sent it right away, so you can probably put it right away.
Okay. Now, an endospore is a structure that some bacteria can basically form in certain conditions. Not all bacteria. Another thing that some bacteria have that not all bacteria will is a flagella. Flagella is actually the plural. It would be flagellum.
Yeah, and it's a tail.
So here, this bacteria has two flagella off the side of it, and they are used for movement. We may have talked about this
Do
before.
sperm cells have them or
Sperm
something?
cells do have a flagellum.
Millions.
Now they're not going to use these to walk around or anything. They will move them in a corkscrew pattern, and if they're in a liquid environment, that'll propel them around the liquid environment. So some bacteria are able to move around using their flagella, or flagella. In this case, he's got two. Usually you don't see more than two. Okay, some bacteria also have little structures that stick off to the side called pili.
What's his last name?
Okay, the pili, or not pili, it's pili.
Yeah,
P-I-L-L-I.
Philadelphia.
It's an I on the end.
Yeah, what's it called?
Okay? The point of the pili is they work not unlike Velcro. When something bumps up against it, they can use it to attach to that thing. Such as pizza. So the pili here can be used for attachment. And that attachment could be to, the surface of another organism. It could be to a food source. It could be to each other.
What are those small little ripples around there?
Small little
It's
ripples.
like those dots, like,
I don't know,
yeah, those.
just little things that are sitting in the fluid that bacteria. Okay? Now, bacteria reproduce through binary fission. We've talked about this before. Basically, make a new chromosome, so there's two copies, break in half, you get two identical cells. So overall, process of binary fission is very simple. Why don't they have to go through mitosis? They don't have a nucleus. There's no nucleus, there's no multiple chromosomes to separate. As long as one chromosome ends up in each part of the cell, everything's cool. And they do have a little bit of a system for that, but we're not going to worry about that right now. Okay? So, remember we've talked about the benefits. What's the big benefit to binary fission? Versus,
You
I don't need a mate. Versus sexual reproduction, no mate needed. You want to make a new copy? Bam. You got all the things that you need to. What's the big problem with binary fission?
And all the same.
They're all the same. There's no genetic diversity. If something comes along that kills one, it's going to kill all of them because they're all the same.
They like bananas, right?
What's that?
The banana flag.
Right. Like the banana thing. However, bacteria do have sort of a way around this. They're able to trade genetic material through a process called conjugation.
And I don't actually like it.
Okay, listen. I don't know what the other classes use and how they use it, but in this class, conjugation refers to the transfer of genetic material between two bacteria cells. So basically, one bacteria cell can extend a modified pili and attach to another cell, and then it's going to be able to transfer, copy and transfer some of the genetic material, usually in the form of a plasmid, to another bacteria cell. And if those bacteria cells have some slight differences to them, this is a way of increasing the genetic diversity, getting new genes. And that whole process of getting new genes is called, horizontal transfer. Now they're not replacing their DNA, they're just getting some extra DNA. That DNA may be different enough that it gives them some sort of an advantage. This is basically bacteria cells.
Nice. Yeah.
Okay? How could two bacteria end up different though? Mutations. So you have two bacteria lines, they're going through binary fission, they're creating these identical things, but a mutation in one causes a different genetic change, and then later, those bacteria lines can meet up, that genetic change can be transferred to the other one, horizontal transfer. Now we have this different genetic material that causes variation.
So it's kind of like evolution.
Yeah, it has to do with evolution, absolutely.
So all bacteria start at the same time. It's like,
Yep.
Give a bacteria out the back.
Okay, so horizontal transfer is a way for bacteria to keep some genetic diversity, gain some genetic diversity. And here is a micrograph showing, and you can see how long that modified pili can actually get to transfer that genetic material, showing one bacteria cell transferring genetic material. Okay.
I don't. That's like magic or something. Actually, it doesn't say they're
Well,
being.
not bad.
They always said making babies.
Okay.
I think I heard of the movie.
So, and maybe should I rearrange this a little bit, but we talked about we are able to classify bacteria based on their shape. There's a couple other ways to classify bacteria. One is anaerobic versus aerobic.
One's stretchy, one's not.
Huh? Stretchy?
Oh, no.
No.
I'm thinking of like the exercise.
Aerobic means that they,
Water aerobic.
no. They use oxygen. So aerobic exercise is just an exercise where you're continuously taking in oxygen, your heart rate.
Anything.
Okay?
So sitting is aerobic.
Anaerobic means no oxygen. There are some bacteria that live in environments where they don't use oxygen at all. In fact, some bacteria live in environments where oxygen is actually harmful to them. There are anaerobic bacteria that live in your guts. And they don't need oxygen. If they're exposed to oxygen, they will actually die.
So like if you're having surgery or something, how does that, like if they were to cut you open or something?
Yeah, some of those bacteria are going to die. I mean, it's not going to kill all of them off. There'll be some that are left behind in certain pockets that will have to repopulate. You need those bacteria. They help you break down your food. They're beneficial to you. They're anaerobic. They don't use oxygen. So that's one way that another way that we can classify this. We've got, based on whether they use oxygen or not, we've got their shape.
If they just hold their, how do they get oxygen?
They don't have a breath to hold. There's no lungs right
I
now.
know, but how can they take oxygen in there?
It's not a-
And they just need to drown there.
Yeah.
It drowns them.
It's around the.
Oxygen is a highly reactive element. That's what's good about it for the cellular respiration process that we need to have happen to give us energy. But it's very reactive and it's corrosive in this environment with these bacteria cells. It will degrade them. Okay, another thing that we can use to classify bacteria is something called gram staining. Okay? What they do is they'll take, if you go in and you're sick and they think it's a bacterial infection, they'll take a swab, they'll put it on a petri dish, they'll let those bacteria colonies grow and divide and grow and divide, and then they'll put this certain stain called a gram stain on it, and they'll see what happens. Some bacteria cells will be stained a dark purple. We call those gram positive. Some bacteria cells don't really get stained that much because of the makeup of their cellular wall, and so they come out as a light pink, and we call those gram negative. Doctors will use that information to decide what types of antibiotics they will give you. Some types of antibiotics work really good against gram negative bacteria. Some work really good against gram positive bacteria.
So it worked out more.
Well, in most cases, you're going to have one. This is just a picture that shows both of them so that you can see the differences. Okay? What's the other difference between these bacteria strains? Can you see?
Colony formation.
Huh?
Formation. Some of them are lumps.
No, not so much that. Maybe you guys can't see that.
It looks like they
Yeah,
roll,
it is shape of them.
like
The shape
I
of
can
it.
see Africa.
What shape are the pink ones?
They're round.
Like
Cheerios.
No.
squiggly fish.
Okay, we can't see any.
I guess maybe you guys can't see it from there. If you come up here close, I mean, they're small, but you'll be able to see the pink ones are actually bacillus, which is
Oh, rod.
rod-shaped, right? I just need to look over those things. And like I say, it might be hard for you to see back there. The purple ones are round, so those would be dogs.
You need me to spill that?
No, I don't.
Pockets.
How long does it take?
Seed.
The staining thing doesn't take long, like a couple minutes. But usually, if they're getting a swab from your throat or something like that, it'll take 24 hours to grow up enough of them to be able to.
And then they give you drugs anyway? So you like if you strep throat, they just give you drugs anyway.
So
Like sense.
what they tend to do is, okay, strep throat. We think it's strep throat. We're going to do a thing, but based on how it presents and how it infects you, they're going to go, okay, we're 90% sure this is strep throat, so we're going to throw this antibiotic at you. Now, if that doesn't work, they'll go back and look at the test and go, oh, this is actually a gram. I mean, I'm just giving an example. I don't know which type of staining happens with the strep type of bacteria, but they'll go, oh, this was actually a gram negative. We need to change your antibiotics. So a lot of cases, because people don't want to wait, they'll sort of make an educated guess. That comes down to, but especially if there's an infection that won't go away and they need to delve deeper, then they'll definitely do bacterial colonies and they'll do all these tests. Okay, yeah, that's all we got.